Hi everyone, I am Dr. Amrish Shrivastava, a clinical pharmacologist and a medical practitioner. In spite of advance in pharmacology and medicine, there is a significant gap in translating the knowledge of pharmacology into clinical medicine. I am going to start a lecture series on pharmacology and therapeutics covering the various clinical and therapeutic aspects on the subject. Now let us start with the subject of pharmacology. What does it mean? Pharmacology is the science of drugs. What is a drug? Drug is simply defined as any substance that is used for diagnosis, prevention, and treatment of diseases. It involves drug discovery, invention and development, pharmacogenetics, which is talking about absorption, distribution, metabolism and elimination, pharmacodynamics, that is mechanism of action, pharmacogenomics, that is genetic influence on the drug effect, and drug toxicity and poisoning. Unlike centuries ago, when there were very few medicines, now we have thousands of medicines which are available for various indications. Even for one disease, there are several medicines which are available. And therefore, there is a need to learn the principles of pharmacology and therapeutics. Drugs, as we know, that they are the prime tool for the physicians, and any thoughtless prescription can do significant harm to the patient. The goal should be to minimize the prescription error, and it can be done only by thorough understanding of the drug, the disease, and the patient. Then therapeutics. This therapeutic term is derived from a Greek word called therapeutikos, which means inclined to serve. Therapeutics is a branch of medicine dealing with treatment and care of a patient. Now it includes both treatment and prevention. Commonly, people understand therapeutics only as a treatment part of the disease. So therapeutics is a comprehensive discipline with an integrated approach. It could be a drug treatment or it could be a non-drug treatment. When we say drug treatment, it is just one component of therapeutics. Now, before going further, let us see what are the duties of a prescriber, means what are the duties of a physician. The 5 rights, as normally said, are right drug, right patient, right dose, right route, and right time. So these are the thumb rules for safe medicine for the patient. The use of drug should be restrictive, means there should be judicious use of the drug, and one should always think before prescribing that is it really required. We see a lot of prescriptions of vitamins, minerals, antibiotics, and so many things, painkillers and all, which are prescribed just unnecessary, adding the burden of drug as well as the cost. Another important point to look into is the choice of medicine, and the choice of medicine is done on the basis of risk-benefit analysis. What is risk-benefit analysis? It is balancing the risk of the drug versus benefit of the drug, and it depends on the severity of the disease. Say for example in cancer patients, all the anti-cancer drugs, they have a lot of side effects, but still they are used because there is some hope that they can increase the survival. So serious the disease, higher the risk of the drug. Patient should also be the part of treatment decision. Patient should be told about the drug side effects, its benefits, its other aspects which are simple in simple terms, so that he should know whether this treatment is suitable for him or not. And after prescription, it is important to explain the prescription properly, thoroughly, about the doses, frequency, and duration of treatment. It should not be left to the pharmacist or any other person, because when you communicate with the patient directly, there is a significant confidence which is generated in the patient, and it also enriches the bond between the doctor and the patient. After the prescription is given to the doctor, the treatment should be monitored over a period of time and with follow-up visits. He should be asked about any side effects if he has experienced, any efficacy, whether it is lack of efficacy or good efficacy. And the last but not the least, patient should be explicitly told about termination of drug treatment. Termination of drug treatment is important for both acute therapies as well as for chronic therapies. Say, for example, antibiotics. Antibiotics are prescribed for 7 to 14 days or 10 days depending on the infection. But most of the time the patient may stop the treatment early because he feels that now there is no symptom and he doesn't require a treatment. So he should be told about proper termination of the medicine. A lot of patients of diabetes and hypertension who should continue the treatment almost lifelong, they stop treatment in the middle, considering that now their blood pressure is controlled and their diabetes is absolutely fine. So that is why it is important that when you write a prescription detailing to the patient about termination, when to stop the treatment, is very, very important. The another point which to be considered, that before you write a prescription, you should think about a checklist, you can say, which is basically a therapeutic plan. What should be looked into before writing a prescription? The very first is patient. You should know the patient profile, that is, what is the age, whether he is young or elderly, gender means male or female patients, occupation has got a significant bearing, because if the patient has got a night shift working, then the medicine should be chosen accordingly. Physiological state like pregnancy or lactation, and so this is very important to know the patient profile when you write a prescription. The medical condition, there could be the other medical condition a patient may be suffering, like he may be having asthma, he may be having blood pressure, he can having diabetes, kidney and renal dysfunction and liver dysfunction. So all these conditions, they have the significant bearing on the clinical outcome of the treatment. It is important, as we are told in the medical colleges, medical schools, that what is the drug history, what is the treatment history of the patient. And you should know that, because nowadays a lot of people, they take medicines without prescription. They are taking herbal medicines, they are taking alternative medicines, they are taking OTC product, over the counter without prescription, asking to the pharmacist. So it is important, and one question, that are you taking any treatment or have you taken any treatment, is a very important question to be asked to every patient. And then once you write a medicine, you should keep in your mind what is your expectation regarding the clinical outcome, what we call as prognostic endpoint. What do you think? Is it only a symptomatic relief, or it is also taking care of the disease treatment? And when you talk to the patient about the treatment, his belief, his expectation should also be considered. So all these things which are important to consider before writing a prescription. Now just a word about new drug. What is a new drug? New drug means the drug which is recently marketed or recently launched, or a physician does not have the experience of any drug. Say, for example, the drug might be available since 5-10 years, but if the physician has not prescribed the medicine, for him it is a new drug. So what he should do, he should have a complete knowledge of new drug, knowing its official indications, contraindications, precautions, doses, frequency and duration of use. Then also it is important to know the approved uses as well as off-label uses. There are so many medicines which are prescribed in the indications which are not approved by regulatory agencies, or they are not officially approved. They are primarily based on medical literature or the personal experience. So it is important to know whether the drug which you are prescribing is an approved indication or it is an off-label indication. It is suggested that you should avoid using the drug based on your personal experience. Unless there is scientific documentation, there are uses of the drug published in the peer-reviewed journals, the medicine should not be prescribed. The patient should be briefed about the potential side effects, and last but important is that prescription should be clearly written. If it is handwritten, it should be legible. If it is by typing, then of course this is not an issue. A patient, if we see, just to see some changes in the behavior of the patient. The patient behavior is now, we find, quite different than what we were seen earlier. After the uses of internet and so many other media, the patient is exposed to a lot of information which are inaccurate, unscientific, and he is exposed to a lot of information without proper validation. So he ends up using irrational drugs which are expensive, which are ineffective, and even they can cause a lot of side effects. Another important behavior which we see in the patient is keep changing the doctor for the same illness, what is known as doctor shopping. It means the patient from one doctor to another doctor keeps going and asking about his illness. And because of these, he is more liable to suffer some side effects or some incompatibilities of drug prescription or drug doses. Few things should be borne in mind, that all the drugs are toxic under certain circumstances. No drug is completely safe. This is one. Secondly, people consider that herbal products and nutraceuticals are absolutely fine. There is no risk of the side effect, but they should also remember that these products have greater portion of impurity and it can cause some harm to the patient. All the dietary supplements which are nowadays used are to be of the standard quality in terms of efficacy and safety. So the proper labeling, the label of all the dietary supplements should be checked before buying those products. All the products, whether it is a conventional drug or it is any supplement, ideally should undergo a proper randomized controlled clinical trial to confirm the benefit of the product. Now coming to the aspect of new drug development. What is new drug development? New drug development is starting from absolutely innovative thought. It could be a drug invention. We say drug invention when it is entirely a new molecule entity. It is very extensive procedure, it is very expensive procedure, and it is also risky, because it is not necessary that the product which you choose or the molecule which you choose will finally succeed in the studies or in the market. So there is a high cost of drug development. There is a lot of regulations which are there for the drug by drug authorities, and in spite of all these, there is a low success rate. So it is a very risky process. We can say especially the new molecule entity is a risky process. It takes around 9 to 12 years for a drug to come to the market after the start. There are several $1 billion which are costing to the company for any product which comes in the market. Now the thumb rule is that the medicine should prove if its efficacy. Means it should be evidence-based medicine. And for this, there is a lot of trials, a lot of studies are conducted. Before human, there are a lot of animal studies. I will just brief you in the later slides. And so this is very important. Now, one thing which is important to know, that there is a difference between clinical trial and clinical practice. Because any new drug undergoes a clinical trial before entering into the clinical practice. But they are not the same. Clinical trial is conducted in few thousands of patients, whereas clinical practice is done in millions of patients. Clinical trial is a controlled procedure. It is done under the supervision of the investigator, whereas clinical practice is uncontrolled in the real world situation, and it is not closely supervised by the treating physician. And that is why a lot of medicines which are found effective in the clinical trials, they fail in the clinical practice, and vice versa. And the last aspect which is important for new drug development is the cost or the health economics. Because after spending so many billions of dollars, there is a lot of competition which the company may face, which the product can face, and also the company has to bear the product liability in case of any side effects or any quality issues. So it is a procedure which is having a lot of complexity. New drug development, especially NME, is a complex procedure. Now, drug development starts from identifying the unmet need. Which drug should be developed? Of course, there is a lot of sources, media, surveys, research, market research. They happen to know that what are the unmet need in a particular area. And once the unmet need is identified, there could be simply an improvement in existing therapy. If already the therapy for that particular illness is there, the people can have some improvisation in the existing therapy. We call it as Me Too drugs. Say, for example, omeprazole, which is used for the treatment of acid disorders. The isomer is found as omeprazole, which has got a better absorption and better safety profile, better pharmacodynamics, and a slight improvisation over the existing omeprazole. Then the use of fixed dose combinations, that is FDCs. Now, two drugs which are already available in the market, they are combined together to improve the compliance and also probably to have the additive effect. So such kind of improvisation is also possible for a new drug development. And then there could be formulation changes. The drug which is available in tablet form is made capsule for some reasons. The oral tablet can be made injectables. The tablet which needs to be swallowed is made as chewable tablets. So all such kind of minor modifications in formulation also can be done for a new drug consideration. Serendipitous discovery or serendipity is an accidental discovery where while doing something, some scientific work, the person or the scientist find absolutely new information about the uses of a medicine. There are plenty of examples of serendipitous discovery in medicine. Penicillin, for example, we all know was developed only by accident by Alexander Fleming, by an incident. Nowadays, most of the drugs, they are developed through combinatorial drug designing. We have libraries of chemical libraries and the computer aided drug designing. By this particular methodology, it is possible to screen several thousand molecules on daily basis. After the screening, then with the help of molecular biology, the disease target is identified. It could be a receptor, it could be an enzyme, it could be any particular tissue site where the drug needs to act. And after the disease target identification and with the help of chemical libraries, there is a lead selection and optimization. Lead selection and optimization means several hundred compounds are identified and then one by one they are studied for various properties, their characterization is done, And there are so many laboratory techniques are available, like X-ray crystallography, nuclear magnetic resonance, and so many other lab techniques, advanced techniques, which help in lead selection and optimization. A small visual just to give an idea that the molecule is starting from 10 to 25,000 molecules almost on daily basis. Out of that, only 10 to 20 molecules, they enter into the animal studies. After that, only 5 to 10 may go for phase 1 clinical trial. I will come to the phase of clinical trial in the subsequent slides. Then 2 to 5 can go into phase 2, 2 may go into phase 3, and finally only 1 molecule can succeed in the market. And sometimes even after so many of efforts, finally the molecule may fail at any stage. Like the molecule can successfully pass phase 2, but it can fail in phase 3. It can pass animal studies, but it can fail in the human studies. So at every time, at every moment, or at every step, there is a risk which is involved in the drug development. Now the preclinical development, once the target is identified, our lead is optimized, the target is validated by various laboratory techniques like immunoassay, bioassays, and various biochemical tests. There are several in vitro and in vivo tests which are conducted, and once the target is validated, the molecule enters into the animal studies. Now in the animal studies, the first thing which is done is the toxicity studies, just to see whether the molecule is safe or not. In the toxicity studies in animals, there are several species which are used depending on the molecule and the type of drug. Say rats, rabbit, mice, dogs, or even primates, that is monkeys, also are sometimes used for toxicity studies. The toxicity study can be acute, subacute, or chronic. The acute study is conducted only by giving single injection of the drug in a high dose, and it is observed for if the animal dies or if there is any behavioral changes. The subacute toxicity study is conducted by giving daily injection for 4 to 6 weeks in 2 or 3 species of the animals, usually rat and mice. The chronic toxicity study is conducted over a period of 3 months, 6 months, 9 months, or even 1 year, including animals like dogs or even other big animals. There is also a study of mutagenicity, just to see the mutation potential of the drug. The reproduction toxicity, just to know whether the drug is safe in pregnant condition or not. The rabbit is one of the common animals which is used for reproduction toxicity. And carcinogenicity, just to know the potential of cancer, if the molecule can cause cancer in the long run. And this study almost takes 2 to 3 years from the start. Besides toxicity study, the pharmacokinetics and pharmacodynamic studies are also done in the animals to know that what is the concentration, what is the profile, what is the action and mechanism of action of the drug, how it behaves in the living organism. Then while the preclinical study is going on, or animal studies are going on, the companies are involved in doing work on large-scale synthesis because they will require for clinical trial. They do a lot of stability studies at different temperature and different conditions, and they work on formulation, whether it is oral or injection. If it is injection, whether it is intramuscular or intravenous. If it is oral, whether it is tablet or capsules, and so on and so forth. And once all this information are accumulated, then it is applied to regulatory for doing a clinical trial in the human subjects. And this application is known as IND, that is investigational new drug application. Now after getting the approval of IND, it enters into the clinical trial, and the first phase of clinical trial is called phase 1. which is normally conducted in healthy subjects. It is conducted in the healthy subjects to know the safety of the drug. The main aim or the main objective of phase 1 clinical trial is to know the safety of the drug. And this is assessed by doing different studies like single ascending dose study when only one dose is used in the ascending order. It can be MAD, that is multiple dose study, where multiple doses are given in ascending orders. The safety is studied in the elderly population above 65 years of age. Then the pharmacokinetics is also studied in all these situations. Every time the drug concentration, what is the Cmax, what is the Tmax, and all these parameters are studied in phase 1. During phase 1, the effect of, if it is an oral drug, the food effect is also studied, whether the drug has got, or the food has got any effect on the drug or not. And then there are studies in the females to know the safety, or if there is any gender issue or any gender effect about the safety as well as about the pharmacokinetics. And the phase 1 is always an open label trial when the patient as well as the investigator knows about whether it is a drug or it is a placebo. After the phase 1, the next phase is phase 2 clinical trial, which is conducted in the patients, and it is in 50 to 500 patients with a drug. It is essentially conducted to find out the proper doses, dose finding study. So phase 2, you can consider that it is essentially a dose finding study, that which dose will be effective in the patient. So you monitor the efficacy at different doses and also the safety. This phase 2 clinical trial is a placebo controlled, most of the time it is a placebo controlled clinical trial, which is randomized. It is multi-center, conducted at multiple centers, and it can be a single blind study or it can be a double blind study. The single blind study, when the patient does not know about the treatment. The double blind study, when the patient as well as the investigator both are not aware of the treatment, whether it is a drug or it is a placebo. Then after phase 2, the phase 3 clinical trial, which is conducted in few hundreds through thousands of patients. Now this is an elaborate clinical trial which is a randomized controlled, and usually it is a placebo controlled trial, but it can also be an active control trial where the drug is compared with the existing therapy. And the major aim of phase 3 clinical trial is to see the efficacy of the drug, followed by safety evaluation. In the efficacy, there could be two situations. There are endpoints which are defined for every treatment or every disease. The endpoints could be a surrogate endpoint or it could be a clinical endpoint. Now, surrogate endpoints are essentially, you can say they are biomarkers, or they are the indicators which tells about the efficacy of medicine, whereas clinical endpoints are the final outcome of the disease. For example, the patient for diabetes, the anti-diabetic drugs, whenever they are studied, the effect of the drug on one of the endpoint, which is called HbA1c, is observed, and any reduction in HbA1c is considered to be useful for a diabetes drug. So that is a surrogate endpoint. Or if a drug for hypercholesterolemia is given and there is a reduction of cholesterol, that is a surrogate endpoint. So such kind of endpoints which are more like a marker, they are called surrogate endpoints. The clinical endpoints are to see the actual outcome of the disease or actual impact of the drug on the disease. So surrogate endpoint are usually taken in phase 3 clinical trial because it takes less time to complete the surrogate endpoint, whereas clinical endpoint can be followed after the phase 3 is over and they can continue even after the launch of the drug. Once the phase 3 trial is over, then it is applied as a new drug application for marketing approval. Now, after the drug is approved, it is studied, it undergoes phase 4 clinical trial, which is done in many thousands patients, And the idea of, or the objective of doing post-marketing study is to do the safety of the drug. It is most like a safety assessment, to do the surveillance, to monitor the side effects, to see the patient compliance, and if it is a drug interaction. Because phase 4 clinical trial is mostly done in the real world setting without much controlled condition. Now, after this drug development, there is a pharmacogenomics, which is quite significantly being developed. Now it is being used, though use in the clinical practice is still limited, but it is one of the very important topic which is discussed in pharmacology. Pharmacogenomics is a genetic influence on drug effectiveness and ADR. And currently about 100 molecules or 100 medicines which are approved by FDA, they have got the genetic information in their package insert or product labeling. The use in regular clinical practice is limited due to the infrastructural issues or availability of the resources. Some of the examples of pharmacogenomics, for example, succinylcholine is one of the muscle relaxant which is used in anesthesia. It is metabolized by an enzyme by hydrolysis, but there are certain people, they are genetically having the deficiency of the enzyme, or the enzyme is not typical, and so they do not metabolize, or they cannot metabolize this drug as fast as normal people, and so there is accumulation of this drug and it causes prolonged apnea. There is a respiratory problem, or there can be respiratory arrest. Similarly, there are genetically deficient people with enzyme called G6PD, that is glucose 6-phosphate dehydrogenase, which are susceptible to hemolysis by antimalarial drugs. Isoniazid, which is a treatment for tuberculosis, this drug is metabolized by acetylation, but this process is genetically determined. There are slow acetylators, there are fast acetylators. Like Asians are primarily slow acetylators, where the Western people are mostly fast acetylators. So because of this, the side effect profile is also different. In Asian population, isoniazid causes neuropathy as the main side effect, whereas in the Western population, hepatotoxicity is one of the major side effects with the same drug. Now, when we talk about pharmacology, now there is a significant development in the field of biopharmaceuticals. People call it a drug by genetic engineering. They are proteins which are manufactured or prepared by recombinant DNA technology using the vector in a host cell. These host cell can be bacteria, it can be yeast, or it can be mammalian cells. And these drugs are very specific. They are given by parenteral route, by injections, and they could be an enzyme, it could be a hormone, cytokines, or monoclonal antibodies. They are quite specific in their action. They may have a long half-life, which may allow the dose to be given on weekly or monthly interval, and quite a lot, the toxicity of these molecules are predictable. Just a few words about stem cells. Stem cells are a new form of therapy where the pluripotent cells are used for various diseases. These are the attractive options now being studied in various diseases like erectile dysfunction, diabetes, urinary incontinence, heart disease, and diabetes. Stem cells are experimentally used in various neurological conditions like motor neuron disease, stroke, and so many other conditions. The embryonic stem cells, because stem cells are essentially driven either taken from embryonic source or from adults. the embryonic stem cells are more effective than adult stem cell. However, there are some debate ethical issues which are always discussed on the embryonic stem cells. The another interesting advanced therapy which has come globally is called antisense therapy, where the antisense molecule, which are essentially oligonucleotides, it inhibits the expression of pathogenic genes and thereby blocks the production of disease-causing proteins. These antisense therapies are given by injections. They are distributed primarily in the liver and kidney. Some of the new discoveries are the new products which are recently launched as an antisense therapy are nusinersen for the treatment of spinal muscular atrophy, a very rare disease, and muprimersen for the treatment of hypercholesterolemia. Lastly, coming to the gene therapy. Gene therapy is also a very recent development in the field of pharmacology, where the administration of nucleic acid, which is normally DNA, is given for various genetic disorders. And these nucleic acid, they are transferred through vectors, which are viruses like adenovirus or herpes virus, for carrying these nucleic acid. Gene therapy is essentially used for the diseases without any cure. They are absolutely incurable. Some of the conditions like cystic fibrosis, hypercholesterolemia, and certain cancers, they are treated by gene therapies. Some of the gene therapies are approved in China and European countries. Recently, US has approved one gene therapy called telemogene for the treatment of melanoma, and it is a gene which is using the vector of herpes virus. So we find that there is a lot of advancement besides conventional drug in the field of pharmacology. That is, it is biotechnological products, biopharmaceuticals, there are antisense therapies, there are gene therapies, there are stem cells, and so the knowledge of pharmacology is not just restricted to a conventional medicine, but also much wider than that. Thank you. 37 minutes का है, अभी इसको सुनते हैं क्या है इसके अंदर, अब चाहे जैसे भी आया होगा